In this video, we will discuss about the last main figure of this paper, where they talk about this ABCG transporter that helps in chemoresistance and is also regulated by Sox2. So, how they come up to this ABCG2 transporter? So, from literature, they are able to find out that this ATP binding cassette that are usually present in the cytoplasm or cytosols that exclude many chemotherapeutic drugs out of the cytoplasm. So, and in this laryngeal cells, cancer cells, they are able to see in the literature that it could be the ABCG2 transporter that helps or that plays that role. So first they check whether this ABCG2 transporter is related to the Sox2 or not. Or so for that they first check. Before going to that, they first check whether this ABCG2 expression level varies in the stiff matrices and the soft matrices or not. And if the culture grown on the stiff matrices has lower ABCG2 levels, whereas the cells that are grown on soft matrices should have higher level of ABCG2 complex. And this is what they expect, and this is what they also see using the qPCR, that ABCG2 levels are high in the soft matrices, and also using the Western blot they are able to see that ABCG2 levels are high in the soft matrices. Thereafter, they checked whether ABCG2 and Sox2 are related to each other or not. So for that, they used this Sox2 silenced cells and then check the ABCG2 levels in these Sox2 silenced cells, and they are able to see that mRNA expression levels goes down of this ABCG2 goes down in this Sox2 silenced cells. And they also check this using the Western blot, which further validate that the Sox2 levels is directly related to this ABCG2 expressions. So once they confirm that the relationship between Sox2 and ABCG2, now they wanted to see whether ABCG2 affect this chemoresistance or not. So for that, they first prepared ABCG2 knockdown cells and confirmed them using the qPCR and also using the Western blot. And thereafter, what I did is, so in this panel, see in the qPCR, they shows that Sox2 has been silenced, and through the Western blot, they shows that ABCG2 goes down in the when they used Sox2 silent cells. And in the panel D, they shows the ABCG2 levels when they use ABCG2 silent cells in the qPCR and also in the Western blot. Now they culture the cells in the soft matrices and treat them with different chemotherapeutic drugs that they used in the previous studies also, that is cisplatin and 5-FU. And they are able to see that the apoptosis goes up, or the percentage of the cells that has apoptosis goes up in the case where there is ABCG2 silenced condition is there. And this is true for both cisplatin and 5-FU compared to the case where control siRNA is used. And they further validate using different approach, that is using the CCK assay, where they check the absorbance. The absorbance goes down for the cells that are treated with this ABCG2 silenced RNAs for both cisplatin and 5-FU treated conditions, and the lower level of absorbance means lower viability of the cells. So this indicates, these results overall indicates that the ABCG2 directly affects the chemoresistance ability of the cells, and this ABCG2 is directly related to the Sox2. So from this overall, they are able to from this paper they are able to find out that these ABCG transporters are the one that plays a role in the chemoresistance ability of this laryngeal cancer cells, and by targeting this ABCG2 transporter directly, they can reduce this chemoresistance ability in the soft matrices and can make the cells more drug sensitive. So this is what they conclude, that ABCG2 can be a chemotherapeutic target. But they did not mention it in the result section that it will be a chemotherapeutic target, but this is what I can infer from the results that are presented in their paper. I hope you understood all the results that are presented in this paper that we have discussed, If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description, and you can also uh, comment your questions about these results or their method approaches. I'll try to answer you in the easiest possible manner. That's all for this video. Thank you for your time.